The chemistry of methotrexate and chemotherapy.
National Cancer Institute predicts approximately 38.4% of people will be diagnosed with cancer at some point in their lifetimes. Literally a growing problem, cancerous cells undergo rapid and uninhibited cell division, resulting in the growth of malignant tumors. Chemotherapy, a well-known and common treatment for cancer, utilizes cytotoxic chemicals to stop the spread of and shrink cancerous cells. Developments in chemotherapy include expansions in the array of chemicals used to combat cancer. One such chemical, methotrexate, targets the cell during the S phase of the cell cycle and prevents it from replicating its DNA. Without synthesizing a copy of DNA, the cell cannot divide and thus the cancer cannot spread. Exactly does methotrexate stop cell division? DHFR, or dihydrofolate reductase, is a small enzyme that plays an important role in DNA synthesis. Folate, or more commonly known as vitamin B9, brings carbon atoms to where they are needed for various chemical reactions in your body. It does this in its metabolically active form of tetrahydrofolate. One example of a tetrahydrofolate bringing carbon to a reaction is the reaction that forms thymine bases. After the tetrahydrofolate brings the carbon atoms to the reaction, the new dihydrofolate gets recycled back into tetrahydrofolate. That's where DHFR comes in. Molecule, you can see the NADPH in green and the dihydrofolate in purple that the DHFR enzyme is closely binding together. Without the interference of methotrexate, NADPH donates an electron to dihydrofolate, creating a reduced form of folate, tetrahydrofolate. Tetrahydrofolate is what is needed to synthesize nucleic acids, and here you can see where this hydrogen atom attaches to the tetrahydrofolate from the NADPH, which results in an oxidized form of NAD+ and a reduced form of dihydrofolate, which is then tetrahydrofolate.
Once tetrahydrofolic acid is produced by the DHFR enzyme, it is made into 5,10-methylene tetrahydrofolate by reacting with the amino acid serine. This reaction is catalyzed by the serine hydromethyltransferase enzyme. Products of the reaction include glycine, which is another amino acid, and water. The nitrogen from the tetrahydrofolic acid is reduced, having a positive 5 charge to only a negative 3 charge in glycine. The hydrogen from the serine is oxidized, going from having a negative 1 charge to a positive 1 charge in water. 5,10-methylene tetrahydrofolate then undergoes another redox reaction, reacting with water to produce 10-formyl tetrahydrofolate, along with diatomic hydrogen. The carbon from the 5,10-methylene tetrahydrofolate is oxidized, going from a charge of 0 to positive 2. The nitrogen is reduced, going from a charge of positive 5 to negative 1/3. 10-formyl tetrahydrofolate can then be converted into a host of other biomolecules unique to individual biosynthetic pathways for different amino acids. Without this molecule, amino acid synthesis cannot take place, and thus DNA replication cannot occur.
Methotrexate is an antifolate, meaning it works to antagonize the actions of folic acids, which happens to include the division of cells. The chemical structure of methotrexate closely mimics that of folic acid. This allows methotrexate to bind to the enzyme dihydrofolate reductase, to which folic acid usually binds instead. Recall that dihydrofolate, a folic acid, requires dihydrofolate reductase in order to convert to tetrahydrofolate. Once converted, the tetrahydrofolate would produce various coenzymes, which would in turn play a part in the production of nucleotides and amino acids necessary for DNA synthesis to occur. By taking folic acid's place and binding to dihydrofolate reductase, Methotrexate inhibits dihydrofolate reductase, preventing the chain of events leading up to DNA synthesis. Without synthesizing copies of DNA, cells cannot divide, and thus the growth of cancerous cells comes to a halt. However, disabling the cell's ability to divide by targeting dihydrofolate reductase with methotrexate also means that healthy cells in the body, which normally divide at higher rates, such as hair and bone marrow blood cells, will fail to synthesize DNA and thus fail to divide. This leads to the common symptoms we all see in media, such as hair loss, anemia from a low count of red blood cells, and dry skin in chemotherapy patients. On the positive side, by inhibiting the enzyme dihydrofolate reductase throughout the body, methotrexate stops cancerous cells which metastasized or moved throughout the body from the original location. Like any other drug, methotrexate will not work for every person or could lose total effectiveness in certain people. Similar to bacteria, the survival of cells from the inhibitory effects of methotrexate and other cytotoxic drugs used in chemotherapy could lead to the mutation of resistance to the specific drug and then the multiplication of resistant cells within the body. Additionally, some patients might have genetics which heavily predispose them to cancer, resulting in the reappearance of cancerous cells even after chemotherapy. The future for chemotherapy looks quite bleak as researchers and doctors search for more precise treatments. Chemotherapy works akin to carpet bombing, and so researchers hope to develop a new treatment more like a smart bomb, which will target pathways used specifically by cancerous cells to divide and grow, leaving healthy hair and bone marrow cells out of harm's way.
Methotrexate, as a molecule, has a relatively high melting point of 185 degrees Celsius, or 458.15 degrees Kelvin. This is a result of its ability to form hydrogen bonds, which stands to reason as it is an organic compound. The molecule contains 3 hydrogen bond donors, pictured in black. It also has 6 hydrogen bond acceptors, represented by the gray circles. It also contains many hydrophobic sections, represented by the white open circles. The molecule is thus very insoluble in water. This stands to reason, as the DHFR binding site it interacts with is also proven to be very hydrophobic. Methotrexate was originally known as a methopterin. It was seen as an improved version of the newly discovered chemotherapy drug aminopterin. Aminopterin is another type of folic acid inhibitor, discovered by Dr. Sidney Farber in 1947. Aminopterin was the first drug to cause remission in leukemia patients, and its discovery was a historical milestone in medicine and marked the true beginnings of cancer research. Both drugs work similarly as folate inhibitors. However, methotrexate had a higher remission rate and was quickly favored over aminopterin. Today, it is one of the most commonly used chemotherapy drugs, costing on average 20 cents per dosage. The World Health Organization lists it as one of the world's most essential medicines. Despite its negative side effects, the formulation of methotrexate was a key stepping stone in the ongoing fight against cancer. As previously mentioned, modern research is focused less on treating cancer through carpet-bombing chemicals and more through targeted means. As cancer is a disease caused by the genetic mutations of DNA, many scientists are researching ways to treat or prevent it by means of personalized gene therapies. A thorough understanding of the precursor reactions to the biosynthetic pathways of the cell cycle, as well as knowledge of the chemical properties of the key molecules in this process, are what allowed scientists to make significant breakthroughs in this rapidly advancing field of medical research.